Hello students, today I bring for you a new chapter, and that is Biotechnology Principles and Processes. It is an important chapter and a new unit. In this chapter, we are going to cover important topics like, here you can see, Principles of Biotechnology, Tools of Recombinant DNA Technology, and Processes of Recombinant DNA Technology. We will cover NCERT and this PDF as well. So, let's start this chapter. What is biotechnology? Biotechnology is the technique of using live organisms or their enzymes for products and processes useful to human beings. Then, European Federation of Biotechnology (EFB), full form is asked in exams, defines biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for product and services. Then biotechnology deals with microbes mediated process, that is making curd, bread, wine, etc. In vitro fertilization, test tube baby program, synthesis and using of a gene, preparation of DNA vaccine, and correcting a defective gene. Coming to core techniques of modern biotechnology, it is of two types, genetic engineering and bioprocess engineering. Coming to genetic engineering, the techniques in which genetic materials DNA and RNA is chemically altered and introduced into host organisms to change the phenotype. And the bioprocess engineering says that maintenance of sterile ambience in chemical engineering process for growing desired microbes or eukaryotic cell in a large quantity for the manufacture of antibiotics, vaccines, enzymes, etc. Coming to principles of biotechnology, there are basic steps that are identification of DNA with desirable gene, introduction of the identified DNA into the host cell, then maintenance of introduced DNA in the host and transfer of the DNA to its progeny. First of all, identification of DNA with desirable genes. Traditional hybridization techniques leads to inclusion and multiplication of undesirable genes and desirable genes. In genetic engineering, only desirable genes are introduced, not the undesirable genes. Second step, introduction of identified DNA into the host, that is a vector DNA such as a plasmid is used to deliver an alien piece of DNA into the host organism. Here you can see in this diagram, it is a bacterial cell and bacterial DNA, plasmids are there, and how you can see a vector DNA is produced and recombinant plasmid E. coli, that is host cell, transport cell. Then third is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. That is, piece of alien DNA has no number of the sequence called origin of replication (ORI) needed for starting replication. So it cannot multiply itself in the progeny cells of the organism. Hence, an alien DNA is integrated into the recipient genome. It has origin of replication, it multiplies and inherits along with the host DNA. Then is the process of joining and inserting a foreign piece of DNA into a host organism to produce new genetic combinations is called recombinant DNA technology. First recombinant DNA was produced by Stanley Cohen and Herbert Boyer in 1972. They isolated an antibiotic resistance gene, that is a piece of DNA, from a plasmid of Salmonella typhimurium. It was linked to the plasmid vector and transferred into E. coli. As a result, the gene was expressed and multiplied in the E. coli. Here you can see the diagrams. Next is tools of recombinant DNA technology. Let's see what your NCERT says here. Initially, biotechnology deals with techniques of using live organisms or enzymes from organisms to produce product and processes useful to human beings. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. And the definition is the integration of natural science and organisms, cells, parts thereof, and molecular analog for product and services. Coming to principles, there are two, genetic engineering and bioprocess engineering. Here, Some more points that I highlighted. A sexual reproduction preserves the genetic information while sexual reproduction permits variation. The technique of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Here, in a chromosome, there is a specific DNA sequence called origin of replication, which is responsible for initiating replication. Then, cloning means making multiple identical copies of any template DNA. And plasmid is autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. Then the cutting of DNA at a specific location become possible the discovery of a molecular seizure that is called restriction enzyme. And after cutting, then we have to join that particular DNA. So that is possible by using DNA ligase. Then also you can see after that particular DNA that has to be replicate in the new host cell by using DNA polymerase enzyme and making multiple copies. Here you can see there are three basic steps in genetic modified organisms, that is identification of DNA with desirable genes, introduction of identified DNA into the host and maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Then tools of recombinant DNA technology. Let's see the PDF here. First of all, restriction enzyme, that is molecular seizures, cloning vector and competent host. First of all, restriction enzyme, that is molecular seizures. These are the enzymes which cut DNA at a specific sites into fragments. They belongs to a class of enzyme called nuclease. Here you can see how it cut part in a proper way, proper fashion, proper manner. Then in 1963, two enzymes responsible for restricting growth of bacteriophage in E. coli were isolated. One enzyme added methyl group to the DNA, the other, that is restriction endonuclease, that is cuts DNA. Till now we have more than 900 restriction enzymes have been isolated from over 230 strains of bacteria. Then naming of restriction enzyme. Here you can see it is restriction enzyme E-core1. First letter indicates genus, second two letter indicate the species of the prokaryotic cell from which they were isolated. Example, E-core1 comes from E. coli, RY and 13. R means strain. Roman number, the order in which the enzymes were isolated from the strain of the bacteria. Here, types of restriction enzymes. First of all, exonuclease. They remove nucleotides from the ends of the DNA, exonuclease, and then endonuclease. They cut at specific positions within the DNA. You can see this diagram, it is also in your NCERT. Then they bind to specific recognition sequence of the DNA and cut the two strands at a specific point. Here you can see it is a vector DNA. The enzyme cut both DNA strands at the same site. It is a vector DNA. And E-core1 cuts the DNA between the bases G and A only. Remember, E-core1, it cuts the DNA between the bases of G and A only. So it have to find that particular sequence, then it goes for a cut. When the sequence is GAATTC is there, it's present in the DNA. You can see here GAATTC is present here like this side also. It is a vector DNA. It is a foreign DNA. This portion we have to add in the vector DNA. Okay. So first we have to use both the particular E-core1 for specific cutting. Like this, here you can see this portion is cut out like this, and here also this portion is cut out like this. and then these two are joined together. DNA fragments join at the sticky end, and the joining process done by DNA ligase. DNA ligase. Then it is the recombinant DNA is formed. Then first resident endonuclease is Hind2, which is the first discovered. Hind2. It cuts DNA by recognizing a specific sequence of 6 base pair. This is called the recognition sequence for Hind2. Here you can see GT, then pyrimidine and purine AC. Here also CA, purine, pyrimidine, T and G. So Hind2 cuts like this. Okay. Here some more examples are there. They cut the particular at a recognition sequence. Here ALE1, HAE3, BAM-H1, Hind3, ECOR1. ALE1 and HAE3 produce blunt end. They produce blunt end, even including Hind2 also. Hind2 also they produce blunt end like this. If it's cut like this, here you can see. If it cut like this, then it produce blunt end. Okay, like this. And BAM-H1, Hind3 and ECOR1 produce sticky ends because they cut like this. This one. See? So this cut produce sticky ends, and this one called blunt end. Remember. Then restriction endonuclease recognizes specific palindromic nucleotide sequence. It is a sequence of a base pair that reads the same on two strands, that is 5' to 3' and 3' to 5' direction. Here you can see palindromic sequence means GAA-TTC, GAA-TTC and CTT-AAG. These are the called palindromic sequences. So this particular recognition endonuclease, they recognizes a specific palindromic sequence, then they go for a cut. It means, here you can see palindromes means, for example, I. If you read I in this direction, same letters are coming. If you read I in this direction, same letters are coming. Okay. Also in your NCERT, another one is given, that is Malayalam. Okay. Next, restriction enzymes cut the strand a little away from the center of palindrome sites. Remember, NCERT says all these lines are NCERT lines. It cut the strand a little away from the center of palindrome sites, but between the same two bases on the opposite strands. This leaves single strand overhanging stretches at the end. They are called sticky ends. Like this, sticky ends formed. Then sticky ends form hydrogen bonds with their complementary cut counterparts. This stickiness facilitates action of DNA ligase. As I told you, DNA ligase is there to joining them. When cut by the same restriction enzyme, the DNA fragments have the same kind of sticky ends. They are joining by DNA ligase. Then same points are there, but here another diagram is there. Here you can see foreign DNA and vector DNA, that is plasmid. We cut using both the DNA, we cut by using same restriction enzyme. If it is different, then the size of the DNA cut piece will be not similar with this portion. Then what happened? Ligase by joining foreign DNA to plasmid, where first of all we cut them, both of them. Then I have to join this portion in this particular region. Here you can see blue color is given. It is a recombinant DNA molecule. Then it goes to transformation and in a cloning host, that is E. coli. Then E. coli divides and gives us our particular, for example, this particular DNA. This foreign DNA, it produces a special type of antibiotic. antibiotic. Okay, but E. coli does not produce this type of antibiotic. So for that we have to insert this particular DNA into an host, that is a vector DNA, and then that is a plasmid, and then it in transfer to the E. coli bacteria. After transformation, then the E. coli divides. When it divides, then E. coli able to produce this type of antibiotic because this DNA is given to him. Okay. Next, second is cloning vector. It is a DNA molecule that can carry a foreign DNA segment and replicate inside the host cell. Example: plasmids, bacteriophage, etc. Cloning vector means, for example, we already know what are vectors. Like for malaria, that is the female anopheles mosquito is a vector, working like a vector. So here also it is a DNA molecule that can carry a foreign DNA segment and replicate inside the host cell. Example: plasmids, bacteriophages. Then plasmids are autonomously replicating circular extra chromosomal DNA of bacteria. Some plasmids have only 1 to 2 copies per cell, other have 15 to 100 copies per cell. Then bacteriophages, that is high number per cell, have very high copy number of their genome with the bacterial cell. When the cloning vector are multiplied in the host, the linked piece of DNA is also multiplied to the numbers of equal to the copy number of the vectors. Then features required for cloning into a vector. First of all, in a cloning vector, these are the features are required. Origin of replication, it must be present here, origin of replication. Selectable marker, that is marker gene. Then cloning sites. Then vector for cloning genes in plants and animals. Here for E. coli, cloning vector will be PBR322. Restriction sites are N3, ECOR1, BAMH1, SAL1, PVU2, PST1 and CLA1. Antibody resistance genes are AMPR, that is ampicillin and tetracycline. ROP is given here. ROP means it codes for the protein involved in the replication of the plasmid. ROP. Then coming to origin of replication. It is a sequence where the replication starts. That's why origin of replication is there. A piece of DNA linked to origin of replication that can replicate within the host cell. It also controls the copy number of linked DNA. So for getting many copies of target DNA, it should be cloned in a vector whose origin supports high copy number. That's why we choose PBR322. Then coming to selectable marker, that is marker gene. It is a gene that helps to select transformant and eliminate the non-transformant. That is, if a piece of DNA is introduced in a host bacterium, it is called transformation. Such bacterium is called transformant. If transformation does not take place, it is called non-transformant. Then selectable markers of E. coli includes gene encoding resistance to antibiotics like ampicillin, chloramphenol and tetracycline, kanamycin, etc. Normal E. coli cells have no resistance against these antibiotics. But if we comes in contact with the cloning vector, and which is the PBR322, then these antibiotics are present: ampicillin, chloramphenicol, tetracycline, kanamycin. Then third is the cloning site. To link the alien DNA, the vector needs a single or a very few recognition sites for the restriction enzyme. More than one recognition site generates several fragments and it complicates the gene cloning. That's why it needs a single and very few recognition sites. Then ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. Example: ligation of foreign DNA at BAMH1 site. Ligation of foreign DNA at a BAMH1 site of tetracyclic resistance gene in vector PBR322. As a result, recombinant plasmid is formed. If ligation does not occur, it is called non-recombinant plasmid. Then when a foreign DNA is inserted with a gene of bacteria, when a foreign DNA is inserted within a gene of a bacteria, that gene is inactivated. It is called insertional inactivation. Here the recombinant plasmid lose tetracycline resistance due to insertion of foreign DNA. When the plasmid are introduced into E. coli cells, three types of cells are obtained, that are non-transformants. They have no plasmid, so they are not resistant to either tetracycline or ampicillin. Second is transformants with non-recombinant plasmid. The resistance to both tetracycline and ampicillin. And third is the transformant with recombinant plasmid. They are resistant only to ampicillin. Then here recombinant plasmid can be selected out from non-recombinant one by platting the transformant on ampicillin medium. Then the transformant are transferred on tetracycline medium. The recombinant grow in ampicillin medium but not on tetracycline medium, but non-recombinants grow on medium containing both the antibiotics. Here you can see in this diagram, it is the recombinant DNA, tetracycline gene inactive, and here also tetracycline gene active. It is non-recombinant. E. coli cells, we give transformation plate on ampicillin containing medium. Bacterial colony is formed, it is the master plate, and replica plating on tetracycline medium. Here recombinant, non-recombinant are there. Then thus one antibiotic resistance gene helps to select the transformant. The inactivated antibiotic resistance gene helps to select recombinant. But this type of selection of recombinant is a difficult procedure because it needs simultaneous plating on two plates having different antibiotics. So alternative selectivity markers have developed based on their ability to produce color in presence of chromogenic substrate. Here you can see competent E. coli, foreign DNA, it is inserted with LAC-Z and chromogenic substrate is there. White colonies are formed and the plasmid vector, no insert, chromogenic substrate, blue colonies are formed. Then in this, a recombinant DNA is inserted into the coding sequence of an enzyme beta-galactosidase. So the gene is inactivated due to insertion or inactivation. Such colonies do not produce color. They are identified as recombinant colonies or white colonies. Here, if the plasmid in bacteria have no an insert, it gives blue colored colonies in presence of chromogenic substrate. So you can say this is not inserted. Then like this, here you can see blue and white colonies are there. In this, same points are given, diagram is changed. Then vector for cloning genes in plant and animals. Genetic tools of some pathogens is transferred into useful vectors for delivering genes to plants and animals. Example, Agrobacterium tumefaciens. It is a pathogen of many dicot plants and retroviruses. Agrobacterium tumefaciens can deliver a piece of DNA, that is tDNA, to transfer normal plant cell into a tumor. This tumor cell produces the chemicals required by the pathogen. Tumor-inducing plasmid of Agrobacterium tumefaciens is modified into a cloning vector which is not pathogenic but can cause the mechanism to deliver genes of interest into the plant. Then see the diagrams. Retroviruses in animals can transfer normal cell into cancerous cell. So they are used to deliver desirable genes into animal cells. Third is competent host for transformation with recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through the cell membrane. So the bacterial cells must be made competent to take up alien DNA or plasmid. For this, bacterial cells are treated with a specific concentration of divalent cation, example calcium. So DNA enters the bacterium through pores in the cell wall. Here you can see it is a non-transformant bacteria. Cell wall is there, bacterial genomic DNA is there. Then CaCl2 treatment. calcium treatment is there, divalent cation. Then competent cell, heat shock given 42 degree for 2 minutes, then plasmid transported into the cell. It becomes transformed bacteria. Here, such cells are incubated with recombinant DNA on ice. Then they are placed briefly at 42 degrees Celsius, that is heat shock, and put them back on the ice. This enables the bacteria to take up the recombinant DNA. Here, other methods to introduce alien DNA into the host cell, that is microinjection. Recombinant DNA is directly injected into the nucleus of an animal cell, that is microinjection. Second, ballistic or gene gun. Cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. This method is suitable for plants. This one is gene gun, as you can see. Directly it goes inside the cells, plant cells. Then disarmed pathogen vectors. When it infected the cell, transfer recombinant DNA into the host. It is disarmed pathogens. And the microinjection. Next topic, process of the recombinant DNA technology. Let's see your NCERT, what it says here. Tools of recombinant DNA technology. And the tools are restriction enzymes, polymerase enzyme, ligase, vectors, and the host organisms. First of all, restriction enzymes. In 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of these added to the methyl group of DNA, while the other the cuts DNA. The latter was called restriction endonuclease. The first restriction endonuclease was HIND2. And then it is found that HIND2 always cuts DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. This specific base sequence is known as recognition sequence for HIND2. Beside HIND2, we have now 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. And coming to the naming of the enzyme, first letter of the name comes from the genus, second two letter comes from the species of the prokaryotic cell, that is E core 1 comes from Escherichia coli RY13, while the R is derived from the name of the strain and Roman number following the names indicating the order in which the enzymes are isolated from the strain of the bacteria. Restriction enzymes belongs to larger class of enzymes called nuclease. There are two kinds, exonuclease and endonuclease. Exonuclease remove nucleotides from the ends of the DNA, but the endonuclease make a cut at specific position within the DNA. Each restriction endonuclease functions by inspecting the length of the DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific point in their sugar phosphate backbone. Each restriction endonuclease recognizes specific palindromic nucleotide sequence in the DNA. Here, this diagram, you already go through it. Here, palindromes are group of letters that form the same word when read both forward and backward. For example, Malayalam. M-A-L-A-Y-A-L-A-M. Malayalam. Here also, if you read from this side, same letters are coming. If you read from this side, opposite side, same letters are coming. That is M-A-L-A-Y-A-L-A-M. Malayalam. These are called palindromes. Here, restriction enzyme cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases of the opposite strands. This leaves single-stranded portion at the end. There are overhanging stretches called sticky ends on each strand. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or genomes. Here, another diagram is there, as you can see. This one also we discussed in that PDF. Then, separation of isolation of DNA fragments. The fragments can be separated by a technique known as gel electrophoresis. We will go through it in the PDF afterwards. DNA fragments are negatively charged molecules. They can be separated by forcing them to move towards to anode under an electric field through a medium or matrix. Nowadays, the most commonly used matrix is agarose gel, which extracted from seaweeds. The DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. Then, the separated DNA fragments can be visualized only after staining the DNA within a compound known as ethidium bromide, followed by exposure to UV radiation. Then, you can see bright orange colored bands on DNA in a ethidium bromide stain gel exposed to UV light. The separated band of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. Next, cloning vector. Some plasmids may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. Then, the following are the features that are required to facilitate cloning into a vector, that is origin of replication, the sequence from where replication starts, and this sequence also is useful for controlling the copy number of the linked DNA. Then selectable marker, that is transformation is a procedure through which a piece of DNA is introduced into a host bacterium. Now, normally the genes encoding resistance to antibiotics such as ampicillin, chloroamphenicol, tetracycline and or kanamycin. Next, cloning sites. Presence of more than one recognition site within a vector will generate several fragments which will complicate the gene cloning. So here you can see, so for that we use the antibiotic resistance genes. Two are there. It is ampicillin and tetracycline. Okay. Next, same points are given. We already discussed about it. Next, selection of recombinant due to inactivation of antibiotic is a cumbersome procedure because it requires simultaneous plotting on two plates having different antibiotics. Therefore, alternate selectable markers have been developed. Here, one is in this recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred as insertional inactivation. This is presence of chromogenic substrate gives blue colored colonies. If the plasmid in the bacteria does not have an insert, and if it is inserted, presence of inserted result in the insertional inactivation of the beta-galactosidase gene, and the colonies do not produce any color and becomes white. So they can easily identified. Then vectors for cloning genes in plant and animals. Here, Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA that transfer normal plant cell into a tumor and direct these tumor cells to produce chemical required by the pathogens. Similarly, retroviruses are there in animals, have the ability to transfer normal cells into cancerous cells. So for that, we use the gene of interest to the human. The tumor inducing plasmid, TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic to the plant but is still able to use the mechanism to deliver genes of our interest into a variety of plants. Similarly, retroviruses have also been disarmed and now used to deliver desirable genes into animal cells. Next, competent host. DNA is a hydrophilic molecule. So for that, we need a particular competent host cell for the transformation. Specific concentration of a divalent cation such as calcium, which increases the efficiency with which the DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, then followed by placing them briefly at 42 degrees, that is called heat shock, for 2 minutes only, then putting them back into the ice. This enables the bacteria to take up the recombinant DNA. This is not only one way to introduce alien DNA into the host cell, Some more methods, that is microinjection. Recombinant DNA is directly injected into the nucleus of the animal cell. You already seen that diagram. Then gene gun or biologistics are there. Then also last method, disarmed pathogens are there. Next, process of recombinant DNA technology. Let's see the PDF here. These are steps. First is isolation of genetic material DNA. Second, cutting of DNA at specific locations. Third, amplification of gene of interest using PCR. Fourth, insertion of recombinant DNA into the host cell. Fifth, obtaining the foreign gene product. And sixth is the downstream processing. First of all, isolation of genetic material or DNA. Treat bacterial cell or plant or animal tissue with enzymes like lysozymes, cellulase and chitinase, etc. The cell is broken, releasing DNA and other macromolecules, that is RNA, proteins, polysaccharides and lipids. Then RNA is removed by treating with ribonuclease, proteins are removed by treating with protease, other molecules are removed by appropriate treatments. When chilled ethanol is added, purified DNA precipitate out as a fine thread in the suspension. It is a practical, it is a major experiment which you are going to do or you already done in your biology lab. So first of all, we have to remove the RNA, then proteins, then other molecules also. Then after that, we use the chilled ethanol to purify DNA precipitate, and then it comes out like fine threads like this. Then purified DNA is incubated with the restriction enzyme. As a result, DNA digests, and then DNA fragments are separated by a technique called gel electrophoresis. You can see here the DNA sample is there, and then restriction enzyme is added which cut the DNA. It cleaves the DNA into smaller segments of various sizes. Then it introduced into the PCR. Sorry, gel electrophoresis. Okay, gel electrophoresis. Here the wells are there, as you can see, small wells are there. Here the DNA are being kept. Then gel electrophoresis showing the migration of undigested and digested set of DNA fragments. Undigested are the largest one, the lane one, and the smallest are digested, as you can see. Here in this diagram you can see, the lane 1, that is this lane is undigested, and the other 2, 3, 4 are digested. And the largest, because as I told you, the electrophoresis is a work like a sieving, like a siever, छलनी जिसको कहते हैं. Okay, so the largest particles comes to this type and the smallest one goes to this portion. The DNA bands are there. Next, DNA is negatively charged, so it moves towards the anode. DNA fragments are separated according to their size through the sieving effect of agarose gel. Small sized fragments move farther. This process is repeated with the vector DNA also. Then DNA fragments can be seen as bright orange colored band when they are stained with ethidium bromide and exposed to UV radiation. You can see this is the diagram. Then DNA bands are cut out from the agarose gel. This is called elution. The cut out gene of interest from the source DNA and cut vector are mixed and ligase is added. It creates a recombinant DNA. As you can see, the vector DNA, gene of interest, ligase is given and recombinant DNA is formed. Then third is the amplification of gene of interest using PCR. PCR is the synthesis of multiple copies of the gene of interest in vitro using two sets of primer and the enzyme DNA polymerase. Primers are small chemically synthesized oligonucleotides that are complementary to the region of DNA. Here you can see PCR machine, thermal cycler. Then amplification of gene of interest using PCR. Here steps of PCR are denaturation, annealing and extension. Denaturation. Here you can see annealing, primary is added as you can see. Then primer grows, extension is there, and then amplified. Here, first of all, denaturation. It is the heating of target DNA, that gene of interest at high temperature is 94 degrees Celsius to separate the DNA strand. 94 degrees Celsius. The here heat is 94 degrees Celsius. Okay, to separate the strands. Strands are separated as you can see. It is the double circular DNA, double stranded DNA, and then here you can see it is separated. Okay, each strand acts as template for DNA synthesis. Sorry. Next, second is annealing. It is joining of two primer at 52 degrees Celsius at the 3' end of the DNA templates. Two primers are joined at the 3' end as you can see. Two primers are joined at the temperature is the 52 degrees Celsius. Remember. Then third is the extension. It is the addition of nucleotide to the primer using a thermostable DNA polymerase called a Taq polymerase. Taq means Thermus aquaticus. It is isolated from a bacterium, Thermus aquaticus. It remains active in high temperature during the denaturation of double-stranded DNA. Through continuation replication, the DNA segment is amplified up to 1 billion copies. This amplified fragment can be used to ligate within a vector for further cloning. Then PCR at a glance. The PCR components, DNA sample, primers, nucleotides, Taq polymerase, mixed buffer, PCR tube. Thermal cycler, PCR cycle occur. Denaturation 95 degrees Celsius, strands separate. Then 55 degree primers binds template, annealing process is called. Then extension is there. At 72 degrees Celsius, synthesis of new strands are occurring. Then, using any method, the ligated DNA is introduced into recipient host cell. They take up DNA from its surrounding. If a recombinant DNA bearing ampicillin resistance gene is transferred into E. coli cell, the host cell becomes ampicillin resistant cells. If the transformant cells are spread on agar plate containing ampicillin, only transformant will grow and untransformed recipient cells will die. Here you can see recombinant DNA, E. coli and transformed cells are there. Here transformed cell, non-transformed cell, nutrition medium and ampicillin. Ampicillin resistance colonies are formed and the non-transformant will die. Then fifth one, obtain the foreign gene product. The ultimate aim of recombinant DNA technology is to produce a desirable protein. If a protein encoding foreign gene is expressed in a heterologous host, it is called a recombinant protein. Here you can see it is a target gene, construction of expression plasmid, mammalian cell expression, insect cell expression, yeast expression, prokaryotic expression, and recombinant protein is formed. Cells with foreign gene can be grown in laboratory. The culture are used to extract desirable protein and purify it by using separation techniques. The cells can also be multiplied in a continuous culture medium. Here the used method is drained out from the one side while the fresh medium is added from the other. It maintains the cells more physiologically active and so produce larger biomass. It yields more desired proteins. Then bioreactors. These are the vessels in which raw material are biologically converted into specific product, enzymes, etc. using microbial, plants, animals or human cells. Bioreactors are used to produce large quantity of products. They can process 100 to 1000 liters of culture. A bioreactor provides optimal growth condition, that is temperature, pH, substrate, salt, vitamins, oxygen to get desired product. Here, the most commonly used bioreactor are a stirring tank reactors. The first one you can see. It is usually cylindrical or with a curved base to facilitate the mixing of the reactor content. The stirrer facilitates even mixing and oxygen availability. Alternatively, air can be bubbled through the reactors. Parts of a bioreactor: an agitator system, an oxygen delivery system, a foam controlled system, temperature controlled system, pH controlled system, and sampling port for periodic withdrawal of the culture. Next, the last one is the downstream processing. Here, it series of processes such as separation and purification of product after the biosynthetic stage. This product is formulated with the suitable preservatives. Such formulation undergoes clinical trial and strict quality control testing. Here you can see upstream processing. Here cell number, oxygen time, RPM, how many degrees Celsius, pH, nutrients and flux rate. Bioreactor and the separator. And here you can see downstream processing, pressure, orifice size, time, then again rate of elution, again time. Here then it goes to this portion, then it comes to like this one, and then again this one, again this one, and concentration unit and the product of variant A. Then the final product is again bioanalysis, safety, efficacy, recovery, cost, then quality control, and after that product comes to the market. Let's see your NCERT what it says. Here process are there: isolation of DNA, fragmentation of DNA by restriction endonuclease, isolation of desired DNA fragments, ligation of DNA fragments into a vector, transferring the recombinant DNA into the host cell, culturing the host cell in a medium at a large scale, and extraction of the desired product. First of all, isolation of genetic material. Here we have to remove the other macromolecules, RNA, proteins, polysaccharides and also lipids. So treating the bacterial cell or plant or animal tissue with enzymes such as lysozymes for bacteria, cellulose for plant cell, chitinase for fungus. The RNA can be removed by treatment with ribonuclease, where the proteins can be removed by treatment with protease. Other molecules can also be removed too. Then that particular DNA can be purified by adding chilled ethanol. Then we can collect the fine threads of the DNA, which is called spooling. Here you can see DNA that separated out can be removed by spooling, like this. If this is called spooling. From this mixture we bring out the whole DNA, purified DNA. It is chilled ethanol. Chilled ethanol. Okay. Next, cutting of DNA at specific locations. Here restriction enzyme digestion are performed by incubating purified DNA molecule with the restriction enzymes. Agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion. DNA, which is a negatively charged molecule, hence it moves towards the positive electrode, that is anode. This process is repeated with the vector DNA also. Then joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with the space are mixed and ligase is added, and this results in the preparation of recombinant DNA. Then amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene of interest is synthesized in vitro using two set of primers. Small chemically synthesized oligonucleotides that are complementary to the region of DNA and the enzyme DNA polymerase. You can see same the steps: denaturation, annealing, extension. First of all, the enzyme extends the primer using the nucleotides provided in the reactions and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of the thermostable DNA polymerase, that is thermus Taq polymerase or thermus aquaticus. Then insert recombinant DNA into the host cell or organisms. Here the amphigen resistance gene in this case is called selective marker. Same steps we already discussed in the PDF. Then obtaining foreign gene products. If any protein encoding gene is expressed in a heterologous host, it is called recombinant protein. Then to produce in large quantities, the development of bioreactors, where large volume 100 to 1000 liters of cultures can be processed, was required. Mostly the bioreactors are stirring type. Here you can see simple stirred tank bioreactor and sparged stirred tank bioreactor, through which the sterile air bubbles are sparged. Then the stirrer facilitated even mixing and oxygen availability throughout the bioreactor, and the bioreactor has agitator system, an oxygen delivery system, a foam control system, a temperature control system, pH control system, and sampling port. Then last one is the downstream processing. It includes separation and purification. You already see it, and with this we have completed this whole chapter. As you can see, downstream processing. So thank you all for watching this video, and if you find this video helpful, then give a like. Do not forget to comment, and new viewers, please subscribe my channel. Thank you. Jai Jagannath.